So, nobody really knows what causes MS. I think that's not debatable. And it's likely that MS is not one single disease, but maybe a group of diseases, which have in common an immune attack on myelin as the final aspect of disease manifestation. And there may well be a number of different primary causes in MS, at least from my perspective. And it's probable that MS only becomes clinically apparent in a certain immune background, people with a certain genetic background. So what I'd like to discuss is a little bit about the history of MS research from my perspective, and talk a little bit about my thoughts of MS during the late 90s, and then end up with Zamboni's findings. And so for much of the research history in MS, there was a focus on blood flow as being a problem. So impaired blood flow had some causal relationship to multiple sclerosis. And this idea essentially died out by the early 60s, when the immune problem became the focus of MS research. And so the thinking that the immune system was involved in MS actually started in the 30s, where it was demonstrated that if you immunize rhesus monkeys with CNS matter, you do end up with MS-like symptoms. And in the 1960s, there was a demonstration that if you administer ACTH, which increases glucocorticoids in the individual, during an MS attack, then you have a remission. So clearly, inflammation plays some role in the clinical symptoms of MS. And the immune system is incredibly complex. And it became an area of study for a selected number of individuals. So it's complex because there are many different cellular players in the immune system, and many of the players look morphologically identical. The only way you can distinguish a CD4 T cell from a CD8 T cell is by using molecular markers. And what increases the complexity is that even within a specific population of cells, for example, T lymphocytes, there are probably 25 million different T lymphocytes in each of us, each of which can recognize a very specific antigen. And recognizes this antigen in the context of a protein complex on the cell membrane known as the HLA antigen, and there are many different variants of this protein complex. And so antigen recognition is a very complicated area to understand. And there are probably as many different types of B cells. B cells produce the antibodies. And what regulates T cell activation or T cell inhibition is very complex and very poorly understood. And so MS became known as an autoimmune disease. And especially in the 70s, and this is when the molecular understanding of immunological interactions began to be understood. But again, these were very incredibly complicated interactions, and only a small group of people claimed to have some understanding of these interactions, and these were known as immunologists. So MS, as now considered an autoimmune disease, as an area of scientific research, was dominated by immunologists. And the neuroscientists played very little role, and mainly because I think the immune system is so complex. So forgotten during this time when immunology as a science really started to develop is all the evidence suggesting that maybe blood flow plays some role in MS. And really, at that time, who could blame the researchers? Because at that time, how could blood flow result in the MS symptoms with the attack of immune cells against the myelin around nerve fibers. And clearly, one thing was certain, that the demyelination that is seen in MS plaques is being driven by activated immune cells. So this represents a normal myelinated axon with normal glial support cells in the central nervous system, and this cartoon represents a demyelinating lesion in progress with activated T lymphocytes, activated astrocytes, activated macrophages and microglia. So it's unequivocal that myelin seems to be being destroyed by activated immune cells infiltrating into the lesion. And furthermore, in recent years, more than 80% of the gene variants that have been associated with increasing the probability of developing MS are all associated with immune function. They are cytokines, for example, that play a role in regulating immune cells. And the general thinking at the time was you would have to be incredibly naive not to consider MS an autoimmune disease. Okay, but the big question is, what initiates this immune attack? And the immunologists have an answer. They say that in the immune activation or activation of immune cells, some factors will cause activation of a particular T cell population, but other factors will inhibit that activation. And so they characterize this in cartoons as having these teeter-totter devices. So if the activator molecules dominate, you have activation of an immune attack, but if the inhibitory molecules dominate, you have an inhibition of the immune attack. And in recent years, the immunologists began to get a more complete understanding of these activation and inhibition pathways. So I won't go through this cartoon, but anything in green would activate that cell. Anything in red would inhibit. And so the immunologists simply say that there is an imbalance between the activating factors and the inhibitory factors. So if this is the case, what's causing that imbalance? Is it simply a disturbance of the regulatory system because of random events in cell signaling, or perhaps triggered by outside factors such as fluctuations in the levels of vitamin D? So this is essentially the view of the immunologist. Or is it a change due to damage of certain regions of the brain and spinal cord which activates that immune attack? So one should always have an open mind in science, and you rarely actually see open-minded scientists because they have a particular perspective and they will pursue that perspective. And now I want to go a little bit into some of my research history. And as Bill mentioned, we were founding members of the Saskatchewan Stroke Research Center, established in 1989. And so my research involved mostly studies in cell culture on astrocytes, one of the brain supporting cells, and oligodendrocytes, another of the cells that produce the myelin, microglia, etc. But I was reading widely in stroke research. And one thing that became clear to me is if one reduced blood flow to the brain in an experimental animal, such as a rat or a mouse, the first damage that one can detect is in white matter. So the white matter seems to be much more susceptible to a reduced blood flow than gray matter. And I had also demonstrated in my research that the oligodendrocytes are very susceptible to disturbances. If you increase temperature by a couple of degrees, you can kill them. If you decrease oxygen supply, you can damage them. So in 1996, colleagues and I organized a symposium, and we invited speakers from all around the world. And the symposium was entitled Cell Biology and Pathology of Myelin. And one of the speakers was Moses Rodriguez, who was a neuropathologist. And he described some of his findings on early MS lesions, and pointing out that he could see in very early lesions myelin damage, damage to the oligodendrocytes, without any immune cell into the environment. Okay? So that made me start to think is perhaps the immunologists are incorrect. There may be some sort of external trigger that activates the immune system, and this may be damage to the oligodendrocytes or the myelin. And so this led me to extensively research the PubMed accessible literature. So PubMed is an electronic library that you can get titles of papers and abstracts, and at the time I was doing this research, it didn't go into the early 60s, so I was completely unaware of the research by people such as Putnam and others who have implicated a reduction in blood flow as being a causal factor in MS. But what stood out in my search was that the first indication of an MS lesion is a change in the blood-brain barrier. And this change could be seen not just in white matter, but also in the retina. And the retina has no white matter. There's no oligodendrocytes, there are no myelin, but you can see these early changes in the blood-brain barrier. And alterations in the blood-brain barrier are necessary for immune cells to invade the brain. Normally they don't invade the brain, but they need specific mechanisms allowing them to cross the blood-brain barrier. So the blood-brain barrier is comprised essentially of the capillaries present in the brain. And this is a cartoon of a capillary. This is a red blood cell. This is brain tissue out here. And the capillaries are lined by flattened cells known as endothelial cells, and in the brain they're unusual, at least in most regions of the brain they're unusual, in that one cell is almost completely welded to the next cell. So nothing can pass, or very few things can pass between the cells from the lumen of the capillary into the brain tissue. Only molecules can pass through the cell itself. Okay? So you needed a change in this blood-brain barrier. And the changes in the blood-brain barrier that are necessary are very schematically and simply outlined in this cartoon. This represents these endothelial cells of the capillary. This would be the lumen of the capillary. And in order for a white blood cell to actually infiltrate into brain tissue, we need to have molecules expressed on the endothelial cells which can interact with other molecules on white blood cells to tether them. This can activate the white blood cells to express yet other molecules which can interact with another set of molecules on the endothelial cells to anchor them tightly and initiate the breakdown of this connection between the endothelial cells and the movement of the white blood cells into brain tissue. So it's really very complicated for a white blood cell to get into brain tissue. So you really need to have a change in the blood-brain barrier in order to have white blood cells entering the tissue. So some thoughts that arose in my mind as I was researching the literature is reduced blood flow can increase blood-brain barrier permeability. You can have these changes in the blood-brain barrier if you have low blood supply, low oxygen, low amounts of nutrients. That has been demonstrated in many experimental models, in stroke, for example. Reduced blood flow will cause damage to oligodendrocytes. So the question arose, could the reduced blood flow in white matter lead to myelin damage and blood-brain barrier changes that allow immune cells to enter the CNS and recognize damaged myelin as foreign in individuals whose immune systems are readily upregulated? So this is just an idea. There was no evidence for it. But clearly these ideas were congruent with the findings in the literature, that the earliest changes to be detected in an MS lesion is a change in the blood-brain barrier and damage to myelin and oligodendrocytes. So I hypothesized in that paper that if there's reduced blood flow to regions of white matter in MS patients, you should be able to demonstrate reduced blood flow, even in normal appearing white matter, and this has been subsequently confirmed in a number of studies. I hypothesized that expression of hypoxia-inducible genes, these are genes that are turned on in the presence of low levels of oxygen, should be present in MS plaques, and that has been subsequently confirmed. And the question is there more ready upregulation of aspects of inflammation in MS patients? So we don't really know that, but the results from the clinical trials with dimethyl fumarate to me suggests that maybe this is a possibility that certain aspects of inflammation are more readily upregulated. But these are ideas. So you need to be able to test these ideas. And they sort of went by the wayside, mainly because they went against the dogma that clearly everybody knows MS is an autoimmune disease. So if you look at blood flow, this is a capillary bed. This is a small artery feeding this capillary bed. This is the vein draining the capillary bed. So all the action of importance occurs at the capillary bed. This is where the oxygen diffuses into the tissue, where the nutrients diffuse into the tissue, and where the waste products from the tissue diffuse back into the capillaries. And in my original thinking, I focused on the arterial end, thinking if you have a reduced arterial blood flow, you will have reduced blood flow through the capillaries. It never occurred to me because I thought veins have excess capacity. Veins cannot be involved. You can't have a problem with blood flow through the veins. But I was wrong. It's complete ignorance. I was looking at veins from sort of an anatomical view, from a classical physiological view, where veins are considered to act in part as reservoirs for stored blood. But if you impede blood flow here, you're going to impede blood flow through the capillary bed. So Zamboni had an open mind, had a very innovative way of looking at things, and was a vascular surgeon and had some ideas that veins don't always carry the blood they should be. And I should have known this because I was familiar with varicose veins, for example. So he started publishing. The earliest I could find was 2007, I think, bringing this, or maybe a little earlier, and created a huge controversy in the neurological community. And so my thoughts on Zamboni is reduced white matter blood flow a contributing factor in MS? It's congruent with the hypothesis I had earlier. Only properly controlled clinical trials can give really a clear answer that will convince neurologists and rehab physicians or others who are treating MS patients. And the current evidences, which the neurologists would consider to be anecdotal, but there are so many websites now and blogs where patients are talking about their improvements following vein dilation. We should also keep in mind, if it is a contributing factor, and Mark mentioned this earlier, may be so, well, perhaps he didn't quite mention this, it may be so only in certain subsets of MS patients. And the final thought is CCSVI might be a component of disease progression in MS and not play a primary causal role, but plays a role in exacerbating the disease. And normalizing venous return may alleviate many symptoms, but may not make MS as such disappear. But of course, alleviation of many symptoms is very desired. And one thing is clear, that the strategies aimed at modulating the immune system have only modest effects on the progression of MS disease. So just this alone should cause neurologists to have a little more open mind and consider the developing clinical trials to look at seeing whether venous obstruction plays a role in the development of MS, if only in a subset of patients. I think this is my final slide. So if reduced white matter blood flow is a factor in MS, it may only be a factor in individuals with specific genetic backgrounds. In this case, the presence of reduced white matter blood flow in individuals that do not have MS does not mean that reduced white matter blood flow plays no role in MS. And it's likely that MS is a multifactorial disease, and I've diagrammed that by these three overlapping circles. Perhaps you need condition A, condition B, and condition C to have MS symptoms being manifested. And so just because you can have condition B, such as restricted white matter blood flow, in individuals that apparently are neurologically normal doesn't necessarily mean it plays no role in MS. And I thank you for your attention.